Hello and welcome to Nikolay's genetics lessons, and today's problem is the people living in a small town were tested to determine the Rh factor from the 899 samples tested with anti-Rh serum. 75.9% were positive and 24.1% were negative. Assuming a single pair of alleles R, dominant R and recessive R, what proportion of the students would be expected to be homozygous dominant, heterozygous, and homozygous recessive? First of all, I want to tell a few words about Rh blood group. If we find on the surface of red blood cells this surface protein, we say that this person would be Rh positive. But if we don't find such protein, we say that this person is Rh negative. And another question, can two Rh positive people may have Rh negative child? Take a look. So people can belong to the three genotypes, with a homozygous dominant, with a heterozygous, or homozygous recessive. And if, say, two people who are heterozygous but both would be Rh positive, so in their progeny we can expect following genotypes: positive, 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 negative, positive, negative, and negative, negative. So these people would be Rh positive, and this would be Rh negative. So two Rh positive people, as you see, may have Rh negative child. So as you see, plus basically means that protein product would be produced, and we can find on the surface of the red blood cell a person of this genotype on both homologous chromosomes have normal gene that produce normal protein. For example, person who is heterozygous on one chromosome has normal gene, another one defective that doesn't produce this protein or instructions for production of this protein in the form of messenger RNA, and person who has two chromosomes with defective gene on each of them wouldn't be able to produce this protein at all. So we say that this person is Rh negative. According to our problem, we ask for these three genotypes, so we also can use letters. For plus we can use capital R, and for the minus we can use small r. So this is going to be small r genotype. Again, in order to solve this problem, we are going to use Hardy-Weinberg formula, where frequency of the three genotypes, which can be produced by two alleles, and as you see here we have only two alleles which produce these three genotypes, but only two alleles, dominant and recessive. So frequency would equal to p squared plus 2pq plus q squared, and all this would equal to 1 or 100%. And for the p squared we have homozygous dominant genotype, which is going to be capital R. For 2pq we have capital R, small r, and for q squared, I forgot to put squared here, we have small r genotype. And these two genotypes would make one phenotype, which is going to be Rh positive, and this genotype here would make another phenotype, which is going to be Rh negative. Do we need in our calculations this number? No, we don't need it, but this number just tells us that statistically these percentages have to be very realistic, because the sample is large enough. For example, if our sample would be 90 people instead of almost 900, then these numbers can deviate. And if, for example, we will test only 9 people, these numbers wouldn't be reliable at all. So the bigger the sample, the better. So what we know that people who belong to Rh negative group is 24.1%, So 24.1%. That means that other people who belong to the homozygous dominant, heterozygous genotype, both of them make one phenotype, Rh positive, would be, as you see, we already given this number, 75.9%. If we add these two numbers, we are going to get 100%. Now, in order to find the frequency of each genotype, we have to find frequency, first of all, of the recessive allele, because here genotypes equal phenotype. For example, here we don't know if we take person who is Rh positive, whether this person is homozygous dominant or heterozygous. They both would have the same dominant phenotype. So we are going to use this genotype and phenotype, because here phenotype equals genotype. And here we have frequency. And by the way, because we are doing all statistical calculations on the scale between 0 and 1, this is going to be 0.241, and this number here is going to be 0.759. So when we add these two numbers, we are going to get 1, or 100%. Now we have to find frequency of Q, or frequency of the recessive R allele, which is going to be equal a square root of this number, so 0.241, which is going to be 0.491. And what is going to be a frequency of the allele P, or dominant allele R? It is very easy. Just we have to deduct 1 minus 0.491, and we are going to get 0.509. One more time, we have only two alleles, P and Q, or dominant R and recessive R. Only two alleles make three genotypes. So if we know frequency of the recessive allele, we also can say frequency of the dominant allele, because these two alleles should make 100% or 1. So if we know frequency of the recessive, we know frequency of the dominant. 1 minus this number is going to give us frequency of the dominant allele. Now we have to find frequency of the homozygous dominant genotype, which is going to be P squared. And we know frequency of the P, or dominant R allele, which is 0.509, 0.509 squared. And the frequency is going to be 0.259. Now let's find the frequency of the heterozygous genotype, who is going to show dominant phenotype, but would have recessive allele. According to our formula, 2pq equals to the heterozygous genotype, capital R and small r. So, and this one is capital R, capital R. So we have to multiply 2 by frequency of the dominant allele, which is 0.509, and by frequency of the recessive allele, which is 0.491. And the frequency is going to be 0.500. Now let's review our question. What proportion of the students would be expected to be homozygous dominant, heterozygous, and homozygous recessive? So this is going to be frequency of the homozygous dominant genotype. But if you need an answer in percent form, we have to multiply this number by 100. So you remember, we are doing all our calculations on the scale between 0 and 1. But if we need an answer in percent form, we have to multiply by 100, and that means we have to move this decimal point 2 places to the right, and the answer is going to be 25.9%. Heterozygous genotype, we can give an answer like this, or we also can say, again, we multiply by 100, and this is going to be 50%. And frequency of the homozygous recessive genotype we already given 24.1%, or on the scale between 0 and 1 it's going to be 0.241. And this is all for today. Subscribe and see you in the next video. Goodbye.